Hey guys, welcome to our channel. So we're through decoding Manwich. We got everything we need out of it. But we discovered that these conversions and calculations would go on infinitely. I did a whole video on this. Here's your fingerprint, MN. That could convert to 1314, 1314. M is 13 and N is 14 in the alphabetic order. You could then convert that right back to number letters and convert those letters right back into numbers using multiple charts. By the way, we only use the alphabetic order, and down here we use the numerology chart. But you could use the alphabetic order in reverse. That's a thing. You could use this in reverse down here, and this would go on infinitely, and you could get infinite calculations out of it, and eventually you could put these letters and numbers together and go through the categories. Apparently, I was fairly sure, but now I know it's going through the categories. And you could also write equations. Whoops, that's STC. Joan Jett and the Blackhearts, I love rock and roll. I didn't mean to do that though. But eventually you could write equations. And that's just a little example. But I don't have that sort of a math background. I can do simple algebra and basic math. But since it goes on infinitely, we had to pick a finite place to stop, and we stopped here. And on this side, we stopped here. And that gave us, after we paired them off as letters and numbers, MN13, AC14, MA45, DN41, MN32, DM25, VE14, and W23. Now, mind you, this could be run in multiple orders, but we had to pick one. All right. And we got this to go through the stock market and finance, and we got it to go forward through genetics, and now we're on backwards in genetics. So a lot of interesting stuff in genetics, actually, as usual. So we'll start with 32W. That's coming back. That's right here. 32W coming back. That's where we're going to start. And complement factor B polymorphism 32W protects against age-related macular degeneration. So 32W deals with age-related macular degeneration. So that's interesting. We got genetic diseases going forward, and now we got one coming back as well. All right, if we move on from that, you have WN4. Previous studies have shown that WN4 was involved in the myogenic fate of somites in the myogenic proliferation and differentiation of skeletal muscle. So WN4 was involved in myogenic fate of somites and proliferation and differentiation of skeletal muscle. All right. All genetic stuff. All right. But if we're going to move on for time's sake, and some of these did have more than one thing going on with them, but I decided these were long enough that I think I left an example of that over here and an example of that over here. All right. But if we move on from that, we have 41E. And It's a region. So let's come back up here. The complete sequences of both the 73A and 78C regions were compared to the D melanogaster genome to determine if the coding region for amylase is presented in both regions to determine the evolutionary mechanism responsible for the observed distribution of the amylase gene or genes. The D pseudo obscura 73A and 78C linkage groups are conserved with the D melanogaster 41E and 53CD regions, respectively. And this deals with genomic regions between Drosophila and melanogaster. So this deals with stuff I don't understand, but it deals with that, and there's 41E right there. It's a region dealing with genetics. So if we move on from that, you have V5. The V5 epitope tag DNA with a 5-foot, I'm guessing, BAMHI and a 3-foot ECOR is sticky. Sticky end was cloned into corresponding site of the PGPD vector to yield the vector of PGPDV5. And this deals with yeast cells. So if we move on from that, you have 2M. Yes, you have B2M gene and A2M gene. And this DMEL/C2M fly-based gene report. So we'll just go with B2M and take a look at that. I think it told me up there, protein coding. This is a protein coding gene. And A2M, want to bet, protein coding gene. What do you know? So you got 2 protein coding genes with 2M. And like I said, there's multiple stuff for some of this. But to save time, we're just going to go through one basically. And I think I left it over here for another example. But if we move on from that, you have D23. And D23, genetic and epigenetic studies of environmental lung disease. And where's my number? D23, it says right here. So, D23 right here, D23. I thought so. So this deals with environmental lung disease. All right. If we move on from that, you have D2. And this is a protein coding gene. DRD2, official full name, dopamine receptor D2. It's a protein coding gene. Another one. All right. If we move on from that, you have 3N, is it? 3N. Yes. We're to right here. And 3N, the production of characterization of recombination between chromosome 3N. So 3N is a chromosome. All right. And apparently 3A is one as well. And it deals with wheat. But we're going to move on from that. It's a chromosome, and we're going to move to M1. How about the M1 gene, which encodes minor core protein, MU2. So M1 is a protein coding gene again. All right. If we move on from that, you have M14. Gene M14, and it protein family protein M14. So it's a protein coding gene again. Another one. If we move on from that, you have ND5 and MT-ND5 gene. Mitochondrially encoded NADH. So this provides instructions for making a protein called NADH. So another protein coding gene. If we move on from that, you have D5. DRD5 gene. The DRD5 gene provides instructions for making a protein called dopamine receptor D5, which is found in the brain. The protein works together with a chemical messenger neurotransmitter called dopamine. All right. So it's another protein coding gene. If we move on from that, you have 4A. And how about TUBB4A gene? Tubulin beta 4A. And it provides instructions for making a protein called beta tubulin. So another protein coding gene. All right. If we move on from that, we have a window that I opened that I didn't use. But seriously, if we move on from that, you have M4. And the M4 gene of murine gamma herpes virus. So M4 deals with the herpes virus. It's a gene. All right. If we move on from that, you have 41C. 41C, genetic and bioinformatic analysis of 41C and the 2R heterochromatin of Drosophila melanogaster. So we had this before, and this deals with this again. So I'm not too sure what that is. We could spend an eternity figuring it out, but we're going to move on to 1C. And this ends up being CACNA1C gene. And down here, it'll show you calcium voltage gated channel subunit alpha 1C. And this gene provides instructions for making one of several calcium channels. So it's a calcium channel coding gene. Not protein this time, but calcium. All right, you move on from that, you have A3. And here you go. You got molecular genetics analysis for the A3 alleles. And this brachydactyly type A3, genetic and rare diseases. So it's a type of genetic disease. And you also have a gene, SNAR-A3 RNA gene. So cancer biology and molecular genetics of A3. So you got a genetic disease here, you got an allele here, and you have an RNA gene. I was going to see if it was a protein coding gene, but I'm not seeing that it necessarily is. But it's definitely a gene. A3 gene, SNAR A3 gene, RNA gene, among the other things. And finally, you have 1MN or NM, 1NM. And that's right here. Sequence results, mRNA sequence results for the third DPF, which show that the expression of 780 genetic differed significantly between the solvent control and the 1NM OHBCP exposure groups. So this deals with RNA sequencing. And that gets you through going forwards and backwards in genetics. That got you through backwards. The previous video got you through forwards. So some interesting stuff in there, some stuff I don't understand necessarily. And yeah, what do you expect, right? We're going to do chemical next. So I'll try to get that out in the coming days. So that's going to be it for this one. So for now, guys, thanks for all your thumbs up, thumbs down when appropriate. Thanks for all your comments, leads, feedback, and subscriptions. And thanks for watching, guys. And for now, you guys have a great rest of your day.